For this video, we will cover how to complete two-trait Punnett squares. And for our class, we will really only worry about two-trait Punnett squares for autosomal traits with Mendelian or true dominance. This simply means that there is one dominant version of the traits and one recessive version. Let's look at Indian corn as an example, and specifically the kernels. Since we have a two-trait cross, we have to look at two separate traits. So let's look at color, purple versus yellow in our corn, and the smoothness of the kernels, smooth versus wrinkly. Now in terms of color, purple is dominant to yellow, and in terms of smoothness, smooth is dominant to wrinkly. Now we need to designate letters to the traits, and since we can really use any letter we want, I'm just going to keep it simple and use A's for the color and B's for the smoothness. The big thing here is that we just need to make sure we use different letters for the two different traits. Now a big A would indicate purple corn, small a indicates yellow corn, big B indicates a smooth surface, and a little b indicates a wrinkly surface. Alrighty, with a bit of that introduction out of the way, let's set up a cross. Let's make it between two individuals that are heterozygous for both traits. So both individuals would be big A, big B, little b. The next thing we then need to do is determine what the possible allele combinations would be in the gametes of each individual. Another way to say this is, what are the possible alleles each can donate to the cross? And again, we are looking at two traits, so we have to have one of each letter in each gamete produced. This means that if we look at the individual on the left, their possible combinations of A's to B's and large to small letters would be big A, big B, big A, little b, little a, big B, little a, little b. And since the individual on the right has the same genotype, this individual would also be able to produce the same set of gametes. Now that we have our potential alleles set up, we can go ahead and make our Punnett square. Since this is a two-trait cross, we have to have a 4x4 grid, and there should be 16 boxes in the middle. I can then go ahead and add the potential alleles to their place on the Punnett square. Note here that above each column and next to each row, there should be one of each letter, an A and B in this case, not just a single letter. This is one of the major errors seen often with two-trait Punnett squares. And now the fun begins. At this point, we are ready to multiply the potential alleles against each other. And note that as we do this, there are some general rules on writing the resulting genotypes. We always maintain our traits in the same order, meaning that A's should be before B's. Note that this rule also trumps the second one. Our second rule is then that we write the dominant allele before the recessive allele, meaning big A's or big B's should be before small a's or small b's. Alrighty, with those rules out of the way, let's go ahead and start multiplying the alleles in our cross. So starting in the top row, going from left to right, we have big A, big B, big B. Next is big A, big B, little b. Third, big A, big B, and lastly, big A, big B, little b. Second row from left to right would be big A, big B, little b. Next, big A, little b. Third, big A, big B, little b. And lastly, big A, little b. Third row from left to right would be big A, big B. Next, big A, big B, little b. Third, little a, big B. And lastly, little a, big B, little b. And our fourth and last row from left to right would be big A, big B, little b. Next, big A, little b. Third, little a, big B, little b. And lastly, little a, little b. All right, with all that mess out of the way, we're technically done filling out the Punnett square. However, there are generally a set of questions that are asked about our Punnett squares. For example, what is the phenotypic ratio of our cross? Here I'm simply asking, how many are dominant for both traits, purple and smooth in this case? Dominant for color, but recessive for smoothness, so purple and wrinkly. Recessive for color, but dominant for smoothness, so yellow and smooth. And lastly, recessive for both traits, so yellow and wrinkly. To determine the number of offspring that are dominant for both, we have to count how many of them have at least one dominant allele for each trait. So at least one big A and one big B. And to not make this take a day and a half going through, let me just circle all the ones that fall under this group. So here we have 9 individuals. Next, we're looking for the ones that are dominant for color and recessive for smoothness. So here they would have to have at least one big A, but has to have 2 small b's. Again, let me circle them, and we have 3 individuals. Then we can try and find the ones that are recessive for color and dominant for smoothness. These would have to have 2 small a's and at least one big B. And if I circle them here, we have another 3 individuals. And lastly, we have the individuals that are recessive for both. These have to have 2 small a's and 2 small b's. In our cross, there's only going to be one of them, so let me circle it real fast. This means that the overall phenotypic ratio for this cross would be 9 to 3 to 3 to 1. A good rule of thumb to making sure that you accounted for all the individuals in the cross is that the numbers combined should add up to 16, since there are 16 boxes in the cross. Alrighty, that was a bit of a mess, but let's reset and ask the next question, which is somehow quite a bit worse. What is the genotypic ratio of the cross? Here I am asking, what is all of the potential allele combinations, A's and B's plus dominant and recessive versions, in the cross? So to outline these, the possible combinations that we have from this cross would be big A, big A, big B, big B. Big A, big A, big B, little b. Big A, big A, little b, little b. Big A, little a, big B, big B. Big A, little a, big B, little b. Big A, little a, little b, little b. Little a, big B, big B. Little a, big B, little b. Little a, little b, little b. Now let's go through and look at the Punnett square to see how many of each that we have. So looking at the square, we have 1, big A, big B, big B. 2, big A, big B, little b. 1, big A, little b, little b. 2, big A, big B, big B. 4, big A, big B, little b. 2, big A, little b, little b. 1, little a, big B, big B. 2, little a, big B, little b. And 1, little a, little b, little b. And that means that our genotypic ratio comes out to be 1 to 2 to 1 to 2 to 4 to 2 to 1 to 2 to 1. Again, if we add up all the numbers together, it should add up to 16. Make sure that as you go through and complete Punnett squares that you follow the basic steps. Figure out what alleles can be passed by each member. Remember to make sure that you get one of each letter in each gamete. Set up the Punnett square, multiply each set of alleles against each other, and note again here, you want to have 4 letters once they're multiplied together. Then complete the genotypic and phenotypic ratios. This concludes a video covering how to complete 2-trait Punnett squares for autosomal traits with Mendelian or true dominance.